So today we discuss about the humoral and cell mediated immunity that comes under serology and immunology unit. So first of all, what is humoral and what is cell mediated immunity? From name it's clear, right? From name it is clear that humoral immunity, the immunity that present from the birth, or that are humoral immunity that our body will fight against the pathogen. Our immunity when fight like skin, like skin. So this all comes under humoral immunity. And cell mediated immunity means the immunity that mediated by cell like B cell, T cell. So in cell mediated, mainly two types of cell will become, that is B and T cell. Clear? What is the humoral and what is the cell mediated immunity? We discuss in this video about the humoral and cell mediated immunity in detail. So definition. So first of all, definition. Immune system. What are the immune system? The immune system, in our body, the system which fight against the infection, that known as the immune system, right? And what comes under this immune system? Which part of our body will fight against the infection? Like cells, tissue and molecules that mediate resistance to infection. So the cells and tissue like pear patches, tonsils, skin, hair, this all comes under our immune system. Clear? Next, immunology. What is immunology? Immunology is the study of the whole immunity. Study of a structure and function of the immune system. This is known as immunology. Next is immunity. Resistance of a host to pathogen and their toxic effect. Immune response. Collective and coordinated response to the introduction of foreign substances in an individual mediated by the cells and molecules of the immune system. Collective and coordinated response to the introduction of foreign substances in an individual mediated by cells and molecules of the immune system. Clear? Role of the immune system. What is the role of immune system? Main role is the defense against microbes. Defense against the microbes. Defense against the growth of tumor cells. Kill growth of tumor cells. Homeostasis. Destruction of abnormal or dead cells. Homeostasis means destruction of all the abnormal or dead cells. This is known as the homeostasis. Immune systems. What comes under immune system? In immune system, our organ system comes organ, cells and molecules. In molecules like cytokinin, lymphokinin, this all are a molecule. Lymphokinin, right? Immune system. First organ that's come under immune system is tonsil, adenoids, thymus, lymph nodes, spleen, pear patches, appendix, lymphatic vessels, bone marrow. What? Tonsils, adenoids, thymus, lymph node, spleen, pear patches, appendix, lymphatic vessels and bone marrow. This all comes under immune system. Okay. Next is lymphocytes. What are the lymphocytes? Lymphocytes are T cell lymphocytes, B cell lymphocytes. When B cells will differentiate, they differentiate into a plasma cells and memory cells, right? When B cell will differentiated, they converted into plasma and memory cell, and plasma cell contain antibody, and memory cell will remember the pathogen for long time. And next, lymphocytes is the natural killer lymphocytes. Next is monocytes, macrophage, granulocytes. In granulocytes is neutrophils, is eosinophils, basophils. Granulocytes contain neutrophils, eosinophils, basophils. Clear? Now third is antibody. Third is molecules. And in molecules what contain? Antibody, complement system, cytokinin, interleukin and interferon. Remember this. These all are a molecule. Antibody, complement system, cytokinin, interleukin and interferon. There are two types of immunity. One is innate immunity and next another is acquired immunity. Innate immunity means the immunity that present from present from our birth, and this immunity is known as a first line of immune response. What? First line of immune response. And it relies on mechanisms that exist before infection. That exist before infection. And next immunity is adaptive or acquired immunity. Acquired immunity means what happened? The immunity and the substance which acquired after birth from the environment, the immunity adapt by that process is known as a acquired immunity. And this adaptive immunity or acquired immunity is known as second line of response. If when innate immunity that present from our birth is fail, after that acquired immunity will produce their effect, their response. So that this immunity is known as second line of response. Acquired immunity relies on mechanisms that adapt after infection. When infection takes place, first innate immunity will be activated. When innate immunity will be activated, and after that the infection will not cure, then artificially we take a drug, right? Or enhance our immune system, we eat something drug like that, so that our immunity will be increased. When antibody will be react as a second line of response, the B cell will be differentiated into two types, that is plasma cells and memory cells. So this memory cell will be always remember this type of immunity for a long time. When second time this same infection will be attack on our body, then immediately the second line of response, that is acquired immunity, will be activated and they kill that pathogen. So this is the mechanism. Acquired immunity handled by T and B cell. Which? T and B cell. One cell determine one antigenic determinant. Remember this. One cell determine one antigenic determinant. So first is innate immunity. Innate immunity based on genetic makeup. Already told you that innate immunity are present from birth. Innate immunity are present from the birth. Relies on already formed components, rapid response within minutes of infection. Innate immunity, what happened? Innate immunity having a rapid response. What? They having rapid response. When infection will be will attack, when infection will attack on a body, the first line of defense is innate immunity. They activated in minutes. It is not a specific. Innate immunity is not a specific immunity. Same molecule say responds to a range of pathogen. Has no memory. Remember this. Has no memory like B cells or B cells. Does not leads to clonal expansion. Does not lead to clonal expansion. Innate immunity mechanism. Mechanical barrier. In mechanical barrier, what present? Skin, acidic pH in stomach, cilia. Humoral mechanism, lysozyme, basic proteins, complement interferons. And in cellular defense mechanism, natural killer cells, neutrophils, macrophages, Mars cells, basophil and eosinophils. These all are a innate immunity mechanism. What? Mechanical barrier, humoral mechanism, cellular defense mechanism. This is the innate immunity mechanism. Clear? Adaptive immunity, second line of response. Based upon resistance acquired during life. What? Based on the resistance that acquired during life, relies on genetic events and cellular growth. Responds more slowly over few days. What? They respond slowly. What? Innate immunity responds fastly within a minute, but this responds in a few days. Is a specific. Each cell responds to a single epitope on an antigen. Responds on a single epitope on an antigen. Has anamnestic memory. Repeated exposure leads to faster and stronger response. First, see, this is a graph. First response is a so late. But second response, see, second response is so fast. This is a latent period. In first response, this latent period is more. But in second response, this is a very less. So, second response is very fast. Leads to clonal expansion. They increase the number. In adaptive immunity, what happened? They increase the number. Adaptive immunity, active and passive. Adaptive immunity, active and passive. They are differentiated into active immunity and passive immunity. In active immunity, they are natural, clinical, subclinical infection, right? In passive, via breast milk and placenta. Passive immunity are provided by the breast milk and placenta. And artificially they are by vaccination, live, killed, purified antigen vaccine. So, these are a artificial type of active immunity. And passive immunity, an artificial type of passive immunity is immune serum, immune cells. What? Immune serum and immune cells. This is the type of passive immunity. Clear? Adaptive immunity mechanism. Cell-mediated immune response. First is cell-mediated immune response, that is T lymphocytes. Next is eliminate intracellular microbes that survive within phagocytes or other infected cells. And next one is humoral immune response. B lymphocytes mediated by antibody eliminate extracellular microbes and their toxin. Remember this, the mechanism of adaptive immunity is mediated by two types, that is cell mediated immune response and humoral immune response. So, cell mediated immune response, that is by a T lymphocytes, and humoral immune response, that is of B lymphocytes, right? So B lymphocyte mediated by antibodies eliminate extracellular microbes and their toxin. Eliminate extracellular microbes and their toxin. Adaptive immunity, that is active and passive. Already I told you active and passive immunity. So first is adaptive immunity. Adaptive immune system has two arms, that is humoral immunity and cell mediated immunity. So in starting I told you that humoral cell mediated immunity is controlled by B and T cells. So that's not, in starting I tell you wrong, that humoral immunity is controlled by B cell and cell mediated immunity is controlled by T cells. Clear? So remember this, humoral immunity controlled by B cells and cell mediated immunity controlled by T cells. Humoral immunity can recognize protein, polysaccharide, phospholipids and nucleic acid antigen. And cell mediated immunity can recognize only protein antigen. Clear? Cell mediated immunity recognize only protein antigen, whereas humoral immunity can recognize protein, polysaccharide, phospholipid and nucleic acid antigen. Humoral immunity can act against soluble or free antigen. Cell mediated immunity recognize antigen presented by APC with class 1 or class 2 MHC molecule. Humoral immunity provides immunity to extracellular bacteria, virus and toxin. Extracellular bacteria, virus and toxin, and causes type 1st, 2nd and 3rd hypersensitivity. Which type of sensitivity they humoral immunity? 1, 2 and 3. Right? And cell mediated immunity provide type 4 hypersensitivity. Okay? And cell mediated immunity cause acute graft rejection. Clear? Now cell mediated immune response. Cell mediated immune response is mediated by T cells, right? So what happened? T cell recognize peptide antigen. What? They recognize the peptide antigen on macrophages in associated with major histocompatibility complex, that is MHC. MHC is of two types, MHC 1 and MHC 2. Actually T cells is of two types, TC cell and TH cell, right? TH is T helper cells and TC cells, right? For these two type of MHC are present, MHC 1 and MHC 2. Identify molecules on cell surface, help body distinguish self from non-self. This MHC distinguish between self and non-self. So T cell goes into effector cell stage, that is able to kill infected cells. T cell goes into effector cells stage, that is able to kill infected cell. This is the T cell activity. See, this is cytotoxic T cell, TC cells. This is the TC cell, right? This is antigen presenting cell, APC, that is called antigen presenting cell. When antigen recognized by antigen presenting cell, this T cell, cytotoxic cell, will came and bind with this, right? In third step, what happened? This helper cell came and bind with this they activate the T cell. They activate the lots of T cells, TC cells, so that they produce lots of molecules. In starting already I told you molecules, cytokinin, antibody, interleukin, this all are comes under molecules, right? So that this when TC cell will be activated, they release lots of molecules, that is cytokinin, and this cytokinin degrades this pathogen. What? They degrades the pathogen. Clear? And this one step is also that they activate the B cell. This helper cell activate the B cell, and this B cell produce antibody, and this antibody degrade the APC molecule, antigen presenting cells. Right? So these are the mechanism of T cells. What? These are the mechanism of T cell. T lymphocytes. T lymphocytes are of two types. Already I told you. T lymphocytes are of two types. That is T helper cell, TH, and T cytotoxic cell. That is TC. This TH cell having CD4 T cells activate phagocytes to kill microbes, and CD8 plus present on TC cells that destroy infected cells containing microbes or microbial protein. They destroy infected cells containing microbes or microbial protein. Now, this is the antigen, right? In antigen, what is are present? Foreign protein, virus, bacteria. What? Foreign protein, virus, and bacteria. This antigen having two types of response. Having two, that is humoral response. They create humoral response or cellular response. If humoral response are present, the B cells will be activated. If cellular response will be present, T cell will be activated, right? When B cell activated, they differentiated into two types. That is plasma cells and memory cells. Another is BM, memory cells, right? Plasma cells produce antibody. They secrete antibody, and this antibody attack on antigen so that they kill that antigen. And next one is cellular response. That is T cells. When T cell recognize, they differentiate into TC and TH cells. TH cells produce cytokinin, or they help to activate B cell, right? And TC cell kill the infected cells. They kill the infected cells by producing the molecule like cytokinin or interleukin like molecule. By that the infected cells will be killed. These are the mechanism of cell mediated immunity. Now, cell mediated immune response. Primary response, production of a specific clones of effector T cells and memory clones. In primary response, what happened? Production of specific clone. The production of a specific clones increases in primary response. Developed in several days, does not limit the infection. Secondary response, more pronounced, faster. Already I told that in primary response, what happened? They take several days for recognizing or fight against that infection. They take several days in primary response or first response. But when memory cells will developed in the body, in second response, they more effective, and they recognize that same infection very quickly. So secondary response is more pronounced, faster, more effective at limiting the infection. An example is cytotoxic reaction against intracellular parasites, delayed hypersensitivity, hypersensitivity, tuberculin test, and allograft rejection. Now, next is humoral immune response. Humoral immune response is a B lymphocyte recognize a specific antigen. B lymphocytes recognize a specific antigen that proliferate and differentiate into antibody secreting plasma cells. What? They proliferate and differentiate into antibody secreting plasma cells. Antibody binds to a specific antigens on microbes, destroy microbes by a specific mechanism. They destroy microbes by a specific mechanism, and some B lymphocytes evolve into the resting state memory cells. What? In resting state memory cell, because B cell will differentiate it into two types, right? In plasma cells and in memory cells. And plasma cells will produce antibody, whereas memory cells recognize the antigen when second time they infect our body system, right? Now, these are the mechanism of the humoral immune system. This is the antigen. These are the antigen, right? This is the antigen specific B cell receptor. Antigen specific B cell receptor class 2 MHC will be activated, right? Class 2 MHC will be activated. They recognize by the class 2 MHC. This antigen is processed via MHC, right? After processing, class 2 MHC and processed antigen are displayed. They activate helper T helper cells. After activating helper T cell, they activate the B cell. When B cell be activated, they differentiated into two types, plasma and memory cells. This plasma cell produce antibody. This antibody kill this antigen. So these are the process or mechanism by which humoral immunity will be takes place. Now, antibody. What are the antibody that are produced by the humoral immunity? So, antibody, also known as immunoglobulin, belongs to the gamma globulin fraction of serum protein. It is a Y-shaped structure or T-shaped polypeptide. They having two identical heavy chain and two identical light chain, and these two heavy chain and light chain are attached by a disulfide bond. All immunoglobulin are not antibody. Remember this. 5 kinds of antibody are present, that is IgA, IgM, IgE, IgD and IgG. IgG having a 70 to 75% of total immunoglobulin and secreted in high quantities in secondary exposure, and this is the only type of immunoglobulin that that cross the placenta, and these immunoglobulin are present before the birth in a child. Major function of this is to neutralize microbes and toxin, opsonize antigen for phagocytosis activate complement system and protect the newborn. IgM secreted initially during primary infection. When first infection attack on a body, then the level of IgM will be increased. But in second time, level of IgG will be increased. Remember this. IgM cannot cross the placenta. Major function: secrete first during primary exposure, activate the complement, used as a marker of recent. And IgA is monomeric in serum, dimeric with secretory component in the lumen of the gastrointestinal tract, in the respiratory tract. Major function: neutralize microbes and toxins. IgD is the monomeric. Major function: present on surface of B lymphocyte, function as membrane receptor. Main role of IgD is unclear. Ig is a hypersensitivity, monomeric. Major function associated with anaphylaxis and play in immunity helminth parasites. Right? Sequential IgM IgG humoral response. IgM is the primary response. IgG is the level of IgG will be increased when in secondary response or produced after the IgM. Higher level persists smaller amount throughout life, produce a larger amount during secondary response. Already I told this. See, this is the graph of IgM and IgG. See, this is the level. In first response, the level of IgM will be increased in first response, and in second response the level of IgG will be increased. Right? Failure of immune response. Immune response help individual defend against microbes. Some cancer immune response can fail. Hypersensitivity reactions, immunodeficiency. Hypersensitivity reaction cause cells damage through excessive immune response to antigen. Hypersensitivity overreaction to infectious agent, allergy, autoimmunity. Clear? Immunodeficiency, loss or inadequate function of various components of the immune system can occur in any parts or state of the immune system. The immunocompromised host has an impaired function of immune system. Immunodeficiency means impaired function of immune system. It is of two types, congenital and acquired. Congenital means primary immunodeficiency and acquired by secondary immunodeficiency. Congenital is genetic abnormality, where acquired is the result from infection, nutritional deficiency or treatment. Cytokinin is a molecule that's secreted by T cells or B cells, right? And they help in destroying the pathogen. Types of cytokines: monokines, lymphokines, interleukin, chemokines, right? So this all about the humoral and cell mediated immunity. So hope you understand this. So I stopped here. Thank you.